Hi, it's so nice to see everyone today. I am just thrilled to be able to announce our speakers today, Dr. Glazer and Dr. Garcia, who are both doctors here at UC Southwestern in the pulmonary clinic. Let me just tell you just a little bit, clinically speaking, I didn't mean that to be a pun, but about them. But Dr. Glazer has been in practice about 20 years. His specialty is really in the occupation and environmental interstitial lung diseases, and he's kind of a puzzle solver, and I assume we'll get some of those puzzle cases today. I know he's spoken about them before, but is really able to look behind the disease and help understand the cause, which of the several hundred interstitial lung diseases a patient may have, and able to get the appropriate treatment for those patients. I think he has a specific, or a very, not specific, but he's well trained in kind of looking behind the curtain, where a lot of other doctors and even pulmonologists don't have that specialty. So we are very lucky to have him here. Dr. Garcia has been in practice about 20 years as well, and her specialty is really in the genetic or familial pulmonary fibrosis. And she has been instrumental in identifying a lot of the rare gene mutations that are associated with idiopathic pulmonary fibrosis. Dr. Garcia is my doctor, in full disclosure. I am incredibly grateful to be one of her patients. I happen to have familial pulmonary fibrosis associated with one of the rare gene mutations that she has helped uncover. I benefit directly from her research. My son Mitchell is here today. I have a daughter, Samantha, as well, who's not here. It will be people like Dr. Garcia and her team that will make a difference for my son and my daughter, because they inherited my genes. And we don't know if they have the gene or not, and it doesn't really matter right now, because they're living good, strong lives, but it's the research and the work of people like Dr. Garcia and Dr. Glazer that will make a difference for people who have the familial, for all of us, but particularly for that familial, that gene mutation, and where can we go from there to help out future generations. So I am humbled and grateful and very happy to introduce you, Dr. Glazer and Garcia, and we'll turn the time over to them.
Thank you for that introduction, and thanks for having us today. I'm going to try to stand over here, because otherwise you'll kind of see the top of my head. So Bill and Shelley asked us to, in some ways, repeat but also update the kind of talks we did a year ago when the PF Warriors group kicked off. And congratulations to you guys on, in such a short time, becoming such a successful and such a large support group. So the way I've structured this talk is, let's go over what's behind the curtain, as she just put it. I'm talking about the different types of pulmonary fibrosis and why it's so important for us to try to tell them apart. And then I'm going to try to get a little technical and go over some of the treatment. So I'm going to show you some of the data that doctors look at, and I'll try to walk you through it. I'm hoping it's not going to be too confusing when you're not used to looking at these types of papers, so I'll do my best, but feel free to stop me if I'm not doing a good enough job and you guys have questions as I'm going through it. And then I'll just say a couple words about some of the ongoing research that's going on. Okay. So to start, one of the reasons that it's so hard on you guys to understand this is the terminology is just so awful. So, the term pulmonary fibrosis, if you just look at it literally, it just means scarring in the lungs. But that's really not the way doctors are using that term. So there's a difference between pulmonary fibrosis and idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis is a specific disease. The term pulmonary fibrosis is just referring to this entire category. It means the same as interstitial lung disease or diffuse lung disease, which are the other terms that doctors use, okay? They all refer to this large group of diseases that affects the deeper structures in the lungs, causing either inflammation, scarring, or both, okay? So I know some of you probably had the experience that you were told, oh, you've got pulmonary fibrosis, and you go looking stuff up and it seems really horrible, and then when you get here you find out, well, I don't have the thing I just spent all this time reading about and freaking myself out about, and it's because of this horrible terminology. So, what I mean by the deep lung, I guess I'll try to use the mouse, but this is your respiratory system. So, when you breathe, there is the mouse. I can't have a mouse, so I'll walk around. So the air comes in here your nose. Because think of it as an upside down tree. For those of you that saw me in clinic, you've heard this before, but this is like the main trunk of the tree. And then it goes out into the big branches that go into each lung. And then the branches keep branching, which is what you're seeing here, just like the branches of a tree, until you get way, 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 way out here where it starts to look like this, and this is it blown up. So these are like the twigs on a tree, the smallest airways in the lung, and then this is, instead of the leaves, you've got these sacs on the end, which are air sacs. So the diseases in this group that the term pulmonary fibrosis or interstitial lung disease is referring to are any of the diseases that affect anything out here. So it may just affect the airways, the twigs, it may fill up these air sacs, or it may just be in the lining of those air sacs right there. That's what we're talking about, okay? You get the symptoms you get because this is where the action happens in the lungs. The air sacs are where the gas exchange occurs. Remember, your lungs, their job is to get oxygen from the air and get it into your body, and to take the carbon dioxide that your body produces and put it back out in the air. And that happens in those air sacs. So when you start having problems down there, you're going to have problems with the oxygen, and it's also going to make it harder to breathe just because of the way the lung gets air in and out of the lungs, okay? So when we look at this group of diseases, there's a couple hundred causes. These causes look different, their prognosis is different, and their treatments are different. So let me just give you a couple examples. So the pictures there, on the top is the CAT scan, on the bottom is the biopsy. That is a disease called sarcoidosis. If I can get this mouse to work, I can't. You don't have a laser, do you? Okay. So there's little dots out on this CAT scan, and those dots equal this on a biopsy, and this is actually a type of inflammation called granulomatous inflammation. So this looks a lot different than this. And this is a different disease in this category. So this is what a CAT scan is supposed to look like, and as you can see up here, it's much grayer. There's that hazy stuff there, and then there's these little black holes. On a biopsy, that gray is because the air sacs are filled up with all these cells. This is called desquamative interstitial pneumonia. It's a different disease. It looks completely different, it acts completely different, it has different causes. That goes to compare that to this. So the CAT scan's on the top, on the bottom, if you look at, so this is the lining of an air sac in this patient. So it's not spidery web thin anymore, it's big, and it's fat, and it's full of scar. But this is called non-specific interstitial pneumonia. Again, the point of this is not to teach you how to differentiate these, it's just to show you how different these things look. And then this one is IPF, or UIP. That's what it looks like, okay? So instead of being the same everywhere, you've got some areas that don't look too bad and other areas that are full of scar, okay? So I think when you see the pictures, it becomes, well, yeah, it does make sense that the prognosis is going to be different and the treatment's going to be different when they look so different, right? The thing that isn't as obvious, though, is even within each of those different patterns, there's different causes of each of those patterns. Your body only can react in a certain number of ways, so there's going to be lots of things that end up leading to the same look on pathology. So just like the different pathologies have different prognoses and different treatment, even within one pathology, the prognosis and treatment may differ depending on the cause, which is why we have to figure out what that cause is, okay? That's why it's crucial. So if we then take a step back, as if, if I had to try to figure out if they were all completely different, we'd probably never figure this out, but fortunately, we can usually group these causes into one of these 5 circles. So sometimes they're caused by medications, which is that red circle. Sometimes they're caused by autoimmune disease, where your body's immune system is attacking yourself. That's diseases like rheumatoid arthritis or lupus, if you've heard of those. That's the green circle. Sometimes it's things you've inhaled, which is the orange circle. Sometimes it's genetics, or it's diseases like sarcoid, which is what the blue circle is. And if we rule all those out, then we're left with the yellow circle, which are the idiopathic interstitial pneumonias. And IPF is one of those, but it's not all of those. It's just one. Okay, so the way we look at this and the way we approach it is we try to always find something from one of the circles on the outside before we're willing to say it's something in the center. Okay, so let me give you an example of a case that we saw. So this was a 48-year-old woman who, when we saw her, had 8 months worth of symptoms with shortness of breath, cough, and fatigue. I know that probably sounds familiar. She had a prior doctor who had sent her in and had told her she has pulmonary fibrosis. That probably sounds familiar to some of you too. She used to smoke. She didn't have any allergies. Her other medical problems were pretty minimal. In terms of exposures, she worked in a warehouse and didn't really have any exposures there, but she did have a bird in her house. On exam, she had crackles, which you guys have all heard about, but she also had something called squeaks, which we think of as a clue because usually those squeaks mean you have involvement of the twigs, those smallest airways. And you usually don't get that when you're talking about an idiopathic interstitial pneumonia. Okay. And she needed oxygen, and her lung function showed that her lungs were smaller than they should be, and the ability of oxygen to get into the blood was reduced. And her prior doctor had done a biopsy, and that had showed that she had non-specific interstitial pneumonia, or NSIP. Okay, this is what her CAT scan looked like. Oh, hit the wrong button, sorry. So, this is kind of the top of her lung, and we'll just look at it going down. And just like that other one I was pointing out, I'll just walk around. So, the color difference is the key. So this is normal, and you see how this is more gray than that? So that's the abnormal for her. It's all that gray. What you don't actually see on her CAT scan is any signs of fibrosis. But this is her lungs as you go down. So at that point, we're left with this decision. You know, do I just call her pulmonary fibrosis, tell her there's not much we can do, or give her some IPF drug? That's what her other doctor wanted to do. Do I say, well, this is non-specific interstitial pneumonia? Well, that would put you in that yellow box, okay? Or do we say, wait a minute, she has a bird? Because birds can cause a disease called hypersensitivity pneumonitis, which can also cause that same finding on path. And that disease, unlike the idiopathic version of non-specific interstitial pneumonia, also affects the twigs. So that fits better for her. So what did we do? We said, get rid of the bird and get your house clean, which we did. And we put her on prednisone and Cellcept to suppress the inflammatory response, because that disease is basically an allergic reaction in your lung, and you'll get inflammation in there. And if the inflammation's there long enough, it can lead to scar. So we got rid of the exposure, or the fuel for the inflammation, and then we tried to put out the fire with the prednisone and the Cellcept. And over the next year, she got a lot better. She came off of oxygen, her physical exam went to normal, her PFTs got a lot better, and this is what her CAT scan looked like. Normal. Someone up front said lucky lady, and you're right. She is, because as a lot of you know, we don't always get this, but we can get this sometimes. And the key to this is that even though she was told she had pulmonary fibrosis, that's a category. She didn't have any fibrosis. She just had the inflammation. Now, this wouldn't have happened if we had said, well, you just have pulmonary fibrosis and we're going to treat you like IPF. And it wouldn't have happened if we just said you have non-specific interstitial pneumonia, because if you don't get rid of the fuel for the fire, even if I'm pouring water on it with the prednisone and Cellcept, she would have gotten worse. Instead, we got rid of both, and we got that. And that's why it's so important for us to figure out what the disease is. Okay? Okay, that's why I picked this case, even though I know it's probably a little frustrating for some of you because we don't have the ability to make you go back to normal. But I wanted to illustrate the point of why it's so important. Because when we look at the universe of all these diseases, there's really actually a wide range of prognosis. Some of them are like this case that have a very good prognosis, and some of them are more serious, like IPF. So on the right over here is the ones that are more scar, because we can't make scar go away, and then as you go towards the left, it becomes more and more inflammation. But even if you stick on the right, and you stick within that fibrotic area, the cause matters for your prognosis. So one of the things, I'm just going to give you an example of that. So one of the things that we want to do as doctors is be able to say, at what point should we refer you for lung transplant? I know some of you here have had a lung transplant. And we have to decide, with help from you obviously, on whether or not you would want it, but we have to decide what is the right time to start thinking about that. Because we don't want to do it too early, because if we do it too early, there is risks with transplant. So we want to make sure the risks of your disease are going to now be higher than the risks of a lung transplant before we do it. So some investigators developed a scoring system called the GAP score. Don't worry about what's in it, but it's a scoring system they developed for IPF to help us predict the mortality of any person's mortality at any point in time. And so other investigators said, hey, maybe we can do this for other causes of pulmonary fibrosis. So they did that study, and what they found is this. So the mortality, or your risk of death, is on the left. So the higher up those lines go, the higher the risk. The top line is IPF. The bottom two lines are fibrotic hypersensitivity pneumonitis or autoimmune disease-related fibrosis. You can see there's a big difference there, even though there's sometimes they have the exact same pathology if we did biopsies. So because of this difference in prognosis, they actually had to put a correction in, which is what this is. This is how they would calculate the GAP score. So the GAP is the G, A, and P, but if they're going to apply this to people without IPF, they have to put a corrector up at the top. The higher the score, the higher the risk, so we have to actually subtract from the score if your fibrosis is caused by something else. So again, even on that fibrotic area of that group of diseases, the diagnosis matters, okay? So how do we make the diagnosis? Well, the gold standard at this point, because the pathology alone doesn't give us a clear answer, is a multidisciplinary discussion. And some of you probably have heard us say we're going to take this to the group and talk about it, or we may have even used those words, but this is what we're doing. So we do it every Monday, and what happens is we have a radiologist who specializes in this, a pathologist who specializes in this, and then the clinical team all come together and we go over stuff. The clinical team provides the clinical information, the radiologist tells us what we're seeing on the CT, and the pathologist goes over the path, and then we fight it out. It's the bottom line. This has been shown when we're all in the same room together, our chances of coming to an agreement and coming to a diagnosis are much higher than if we're not in the room together. And having done it now for a couple of years, it's always because there's some subtle points that don't make it into reports that end up driving what we finally say. Okay, so this is the key to how we make this diagnosis. And I hope I've convinced you why it's so important for us to be able to figure out which of the types it is. So let's switch gears and talk about therapies. So this is where I'm going to start getting technical in some of these slides, okay? First, though, before we start talking about drugs, there's some things that apply across pretty much all of the different types of pulmonary fibrosis. So number one is if there is a potential causative exposure, even if it's only a question that it could have been that, it just doesn't make sense to leave you exposed to it. So we may say, stop that medicine. Are you sorry you have to get rid of your bird? Or yes, you really do have to stop smoking this time. It's not a theoretical risk anymore. So we want to do that. We want to do supportive care things like oxygen, which I heard you guys talking about, and like helping you exercise, because even though it can be hard, it is really important to maintain the quality of life to exercise. We want to treat the other comorbidities that are very common with this, things like reflux or things like sleep apnea, because they can be very common. And then there's always lung transplant, which we always want to think about if everything else isn't working, okay? So from that, we then go to the pharmacologic therapy, and there's a major breakpoint here for our thinking. So even though there's 200 different diseases, and we've got 5 categories, I'm going to leave the genetics out because that's her topic, okay? So excluding that, we end up saying, is this IPF, or is this one of the other ones that features inflammation as part of the pathogenesis, right? Like that case that I showed you. Because we now know that IPF doesn't have any inflammation. You go back even 10, 15 years, we did think IPF was the same pathogenesis. We thought it was fibrosis leading to scar. But instead, we had a series of studies like this. So this is what's called a gene shift study. And what it's doing is it's looking at all of the active genes in the lungs of people that have these various diseases. So on the left is IPF, and the other 2 columns are either that nonspecific interstitial pneumonia or hypersensitivity pneumonitis. And you can see there's a marked difference in the gene activity here. Purple's active, yellow is not. The ones on the bottom are all more inflammatory. That's hypersensitivity pneumonitis. The ones on the top are all related to scar formation and wound healing. It's very different, okay? So this type of research led us to question the way we used to treat ILD. Now this is kind of past history at this time, but I think it's an important point on how we decide on therapy and whether or not and how hard we're going to push to do a biopsy to really know. So that's why I'm going to go over it. But in 2012, a study came out called the Panther trial. And this was a study sponsored by the NIH. It used a consortium of different sites, including us, and it was comparing medicines to suppress the immune system, prednisone and azathioprine. Okay? Azathioprine has been replaced by CellCept most of the time because CellCept's better tolerated more than anything else. It's a little easier for us to use, it's a little easier for you to take, but otherwise we can think of them as about the same. So even though I'm saying azathioprine, we have every reason to believe if it had been prednisone and CellCept, the results would have been the same. And this looked at using those anti-inflammatory drugs in IPF because we were starting to question, should we do it? And this is what it showed: bad things. Okay, so combination therapy means you were on the drugs versus placebo means you were not. And as you can see, the chances of death were worse, the chances of adverse events were worse, the chances of exacerbations were worse, the chances of hospitalization was worse if we had you on anti-inflammatory therapy. Okay, so we don't do that anymore. Not with IPF. But we still need to do that with some of the other diseases, which is why I had the slide before this separated as IPF or the other diseases. And we actually find it's much harder now to decide on when we should do a lung biopsy, because in the past there was a lot of times that we just said, well, we're going to treat them the same, why take the risk? You know, it's a prognosis difference, but it's not going to change our therapy. But now it does. So it becomes a harder decision, and we end up spending a lot of time talking about it. So what do we do when you have IPF, and what do we do when we have the other things, and why do we do it? So I thought I would show you that, and this is where it's going to start getting more technical. So this is the results of the trials for pirfenidone on the left, which is also Esbriet, and nintedanib, which is Ofev on the right. And what you're looking at was the forced vital capacity, which is the measure of the size of your lungs. And it's basically looking at the rate of decline. So the darker line in both is the rate of decline on the medicine versus the lighter one, which is the rate of decline on no medicine. And as you can see, they slow the rate of decline. Okay? That's why we use them. That's why they got FDA approval. Okay? This is why when we talk to you about them up front, we tell you that they don't necessarily make you better, although there are a few people within these studies that it does, but on average, it just slows the rate of decline in your lung function. It doesn't do anything to how you're feeling. It slows the rate of decline in your lung function. Okay? We always thought that because the rate of decline on your lung function is better, that it should eventually affect mortality, because this is a surrogate of saying the natural history of the disease, but we couldn't prove it with these trials. The reason for that is something called power. So if you had a crystal ball And you could look in that crystal ball and say, okay, I know these drugs make people live longer because that's what this perfect crystal ball tells me. You know, other people aren't going to believe your crystal ball, so you're going to have to study this and prove it, okay? So you're going to design some studies to try to prove it so that the FDA says, all right, I believe your crystal ball, and we're going to approve this, this drug. So when they do that, the study has a likelihood of being able to detect that effect that we know is there because of our crystal ball. And that ability to detect it is called power. Okay? So the more uncommon the outcome, the lower the power. So the way you compensate for that is you have to have more people studied or studied for a longer duration. So you can see the problems, right? Longer duration means we probably still wouldn't have these drugs available for you. And more people means it's harder and harder to get the studies done, number one. And both of those approaches lead to a lot more cost. And since the way we do this stuff is pharmaceutical companies end up doing this last step, right? If it costs too much to do, they're more reluctant to do it. That's why they use endpoints like this, okay? So what I want to share with you is some of the new stuff that's coming out about this question and one other one. So the statisticians have come up with a way to address the problem of power. So if the question of power is not enough numbers, what we can do is say, hey, well, there's probably 10 or 12 studies in the literature now on pirfenidone and 5 or 6 on nintedanib all throughout the world. If I combine all those studies, I now have much bigger numbers, and I have much better power. Okay? Now, this isn't as good as doing it directly, because they didn't have the same methods and the same criteria on who gets in and who's not, but it's the best you can do in the absence of doing it from the beginning, okay? That technique is called meta-analysis. Okay? So I'm going to show you the meta-analysis that's coming out, and try to walk you through what this looks like, okay? So when you look at mortality in this meta-analysis, so the way this graph works is you have the dot, which is the estimate of the effect, and the line, so that we know it's going to fall within that range, okay? Does that make sense? This middle thing is zero effect, okay? So if the dot and the lines are all on one side, all on this side, that means it reduces mortality. If the dot and the lines are all on this side, it's going the wrong direction. So prednisone azathioprine, it's over here. What we want is over here, okay? And what this is showing you is that we're getting really close to that. So if you look at pirfenidone, it's right on that line, that edge. When you look at nintedanib, it just inches past that line a little bit. So this is starting to look like these drugs do improve mortality. Is there any difference between them? No. So even though nintedanib isn't quite there yet, it doesn't appear to be different at all from pirfenidone, okay? Can we hang our hat on that from this type of thing? No, this is suggestive, okay? But it looks like they're probably equivalent, and signs are pointing towards what we thought. Now, another technique that we will use to try to improve our power is to, instead of saying we're going to go all the way to mortality, which takes longer and there's smaller numbers, is there any markers along the way that will kind of be what's called a surrogate? So there is one for mortality in IPF, because remember we're talking about IPF therapy here, nothing else, just IPF. So if you have a 10% fall in your forced vital capacity in 6 months, we know that you have a much higher risk of death over the next year. So that 10% fall is a surrogate marker. So how do these drugs look against the surrogate marker? They look pretty good, don't they? Because they're all to the left. The dot and the lines are to the left. And again, no difference between them at all. Okay? So it's looking pretty good. And here's another meta-analysis. The only one I want you to look at on this is here, because this is the mortality. And you can see in this meta-analysis, which was done more recently than the other one, now pirfenidone, it took that thing like this and it turns it upside down.
So this line, it was the line that was in the middle in that other graph, okay? Pirfenidone is now all to the left of that line. Okay, so that looks good. This is not from nintedanib, but if we look at the two of them against each other, again, they're exactly the same and there's no difference. So what does all this mean? It means that we are becoming more and more confident as time goes on that these drugs are going to improve mortality. Okay? We still can't guarantee it, but as more and more data comes up, we're becoming more confident that that's the case, which is good news. Okay? The other question that comes up a lot is, let's say you're on pirfenidone. I'm going to pick that drug because this question has been studied in pirfenidone, but it hasn't been studied yet in OFEV. Okay? But let's say you're on pirfenidone and you have one of those big falls in 6 months. You have that 10% fall. The question then becomes, well, now what do we do? Do we say that the drug didn't work and stop it? Say there's nothing else we can do except send you to transplant? Do we say we should switch you, which I still don't have an answer to? Do we say we should add the other drug to it because they have different mechanisms of action? Good questions. That's not studied yet either, okay? But what we do have information on is, should we just stop and do nothing? Because what we have is this. So this is looking at patients that had that type of decline and then what happened to them over the next 6 months. And if you look at this bottom row, if you stayed on the drug, only 4.5% of those people died over the next 6 months. If you stopped the drug, almost 29% of those people died over the next 6 months. So this is an 84% reduction in mortality over the next 6 months if you stay on the drug. So that's what we recommend at this point, is to stay on the drug. We probably would extrapolate that to OFEV. We don't have any data yet, but everything else looks like the two drugs act exactly the same. And hopefully there will eventually be studies on a switch or the addition of both. Hopefully, to be honest, there'll be a study on the addition on double therapy, even if you didn't have a fall. That requires both drug companies being willing to play ball with each other, and they're not up to that yet. But eventually, hopefully, we will get to that. Okay. So that's where we are on IPF. We have the same two drugs. You saw why we have them, but the data is becoming more and more convincing that they do improve mortality. And at least we know we shouldn't just stop them if you start getting worse. Okay? So let's switch. And let's start talking about these other diseases that feature inflammation. Okay? So I'm going to show you why we pick the drugs that we pick. So in these, like I showed you, all the basic science still looks like if you have hypersensitivity pneumonitis or autoimmune related disease, that it's inflammation that's leading to the scar. It's the immune system that causes that inflammation, so we want to suppress the immune system. Okay? The drugs that we usually use to do that now, like I mentioned before, Cellcept or mycophenolate is the other name for that. And if you're on it, you know. Or prednisone. That's where we usually go first. So this is the data on hypersensitivity pneumonitis. So this is taken from several different centers that looked at their patients and what they did. And the way to interpret this, see the 0 here? That's when the Cellcept was started. Okay? Same thing here. This is looking at the diffusion capacity, which is your ability to get oxygen in your blood. And this is looking at that forced vital capacity. The same thing all those studies on pirfenidone were looking at. Okay? So in this study, the patients all, if we could find an exposure, if they were recommended to remove it, they were all put on prednisone. And despite that, things were getting worse from back here through the initiation. Same thing, getting worse. Then they start the drug. Here, it levels off and goes up a little bit. The dotted line is what would have been predicted to happen based on this slope if we didn't use it. And here's your diffusion capacity where it's pretty easy to see it goes up. Okay? So that's why we use these drugs, is because they seem to work. And for those of you that that are with me in clinic, I will usually tell you if you're on one of these drugs, our goal here is not necessarily to make you better because we can't make scar go away. But if we can get you to stop declining, then that's a treatment. That's a positive treatment effect. Okay, so that's what it shows.
If you look at autoimmune disease, we have a little bit better quality of evidence for one of them, which is scleroderma. So better quality evidence is, instead of looking at like what I just showed you, it's actually doing what's called a randomized controlled trial, the kind of things that got OFEV and nintedanib, OFEV and Esbriet approved. So in scleroderma, there had already been a study that showed that the patients did better if they were on a drug called cyclophosphamide compared to nothing. The problem is that cyclophosphamide can be a very, very toxic medication. So they did a follow-up of that, comparing cyclophosphamide to Cellcept or mycophenolate. This is what you're seeing here. The 0, which is the time point, the start time, is on the left, and you can see what happens to their lung function with both drugs. It goes up. Okay. So again, these drugs work for that. Outside of scleroderma, we don't have randomized controlled trials, but we have studies that are similar to that hypersensitivity pneumonitis trial. So again, this one, the time point is the 0 point is in the middle. On prednisone alone, they were getting worse. They start Cellcept, that line starts going back up. Okay, so this is the data that's out there on why we use these drugs for people that have one of these inflammatory lesions, and it's also why we don't use these drugs if you have IPF. Okay, and that's why we have to tell them apart. Now, unfortunately, that's not everything, right? There's other ones out there, and we still just have no idea what to do with them. Things like asbestos. There's some people that we have to put in a group that we just can't classify what it is, and then there's some rarer ones like that one that I don't even want to say on the bottom, okay? So what do we do with these people? We don't know yet, which is why there's still ongoing research. So, what is the research that's ongoing for IPF? Well, there's still a lot of new agents. I tried to find a slide that I saw Dr. Brown give in a talk. I thought I had it and I don't, because it's really great, because he starts with, this is what we were studying in 2000 or 2002, and it's a blank slide. And it's like, what are we studying now? And there's 15 different things on there. So those studies are still going on, because there's still a lot of room for improvement, right? These drugs that are available have side effects. They don't level you off. They don't reverse anything. And if we could do either of those, how great would that be, right? So there's a lot of room for improvement, which is why there's still new drugs being studied. We are studying 3 of those agents here right now. There's also, finally, a move to look at different endpoints, which is what the Chicago conference that Bill was talking about is about. Because great, your lung function got better, but wouldn't it be nice if we could give you something that made you feel better? Made that awful freaking cough get better, right? Gave you some energy back, things like that. So there are drugs that are now being studied, and people are actively looking at those issues. We've gotten several, the way this starts is they'll send us a request, are you interested in doing this study? And we say yes, and then eventually they'll send us back something to say, okay, we'll send you the protocol if you sign this don't disclose agreement, and then eventually they decide if we're going to be a site or not. So there's several agents out there that are in that phase for cough. And we actually have a study open now for patients with IPF to see if adding a drug to OFEV actually will make you feel better. So I'm very encouraged that we're finally studying that type of endpoint. The reason that the drug companies haven't in the past is the FDA has been resistant to it. So they won't oftentimes approve a medication based on just those quality of life endpoints. So hopefully that is going to start to shift. The other major question that's out there is, what about if you don't have IPF? What if you've got these other diseases that feature fibrosis that we don't know what to do with? Or what if you have hypersensitivity pneumonitis or autoimmune disease and you've got fibrosis? I mean, great, we can give you these medicines to shut down the immune system, but it is fibrosis. Those medicines that are approved, they're not, there's nothing specific per se about fibrosis in IPF, so maybe those medicines will work for those other things too. So there are actually studies on that right now. We are already enrolling for scleroderma, and within a couple of months we'll have a study open on the use of OFEV for some of those other things. Okay. And then of course there's what Dr. Garcia is going to talk about, which is the genes, and what is that going to tell us, and are we going to get any can we derive any other therapies from the genes? Okay. So with that, on behalf of myself and the ILD team, I'd like to thank the PF Warriors, and especially Bill and Shelley, for all the work you guys are putting in. And I want to thank all of you and your families for giving us the opportunity to help you and be part of your care.
I have a question.
Yes.
I know this is very, very complicated, and every situation is different, but in the treatment phase, how much input comes from the patient? You know, I mean, do we have any say in
Of course.
if we can use it or not? Or?
Yeah. So I actually meant to say something about this, and I didn't. So whenever we get to the point of treatment with IPF, the way we decide on the drugs is if you have a contraindication to one or the other, obviously we don't offer you that one. If you don't, we would want your input, and we would want to give you the information about both and let you decide. So I have patients that say, I don't want to take them, and that's their choice. I have other patients that say, I want this one, but I don't ever want that one, and that's their choice. Okay, and the same thing is true on, the prednisone cell set side. If you've got one of those diseases, we have plenty of patients that said, I just don't want that stuff, and that's their choice.
The other question I have too is, when you have those meetings on Monday, does every case that, like, it's here at UT Southwest, does everyone go to that meeting?
Not everyone, because there are times that it's pretty straightforward, and the clinician just talking to the radiologist and the pathologist can easily get to that point. There's always a conversation. It's not always the whole group. So the ones that go to the group are the ones that it isn't crystal clear based on the information leading up to it. If someone has rheumatoid arthritis already and they come to us, I don't have to take them to that board. I know it's the rheumatoid arthritis that caused the lung disease. If they had rheumatoid arthritis and they were exposed to a bird and they're on a drug that could do it, well, then I may take it to that board to say, all right, guys, which one are we thinking this is most likely due to? Okay. Does that make sense?
Yeah.
All right, so let me pass it over to Dr. Garcia, and then we'll both be here for questions after that.
Thank you, Dr. Glazer. So the way we split this up is that Dr. Glazer was going to talk more about diagnosis and treatments, and I'm going to talk a little bit more about research and kind of the newer things that are going on in research on pulmonary fibrosis. And I've also devoted some part of the end to talk about stem cells, because a lot of my patients are coming to me and asking me about this. And this is something Bill and I talked about a little while back, but I definitely want to bring that into the conversation. And I am happy if you interrupt me, if you have a question that you're just dying to get out, because sometimes it's better just to address some questions as we go along. And so feel free to raise your hand. Let me know where we are in terms of our general understanding of the talk. So I'm going to put this in the framework of talking about different types of research. And when we say research, just like pulmonary fibrosis is a big umbrella term for all the different disorders that we are talking about, there's many different types of research. And I know a lot of you have interacted with us and with other individuals and studies in one of a number of different ways. I'm just going to kind of touch upon some of those. One, there's protein or cell-based studies. This is basically deep in the lab. These are the scientists that are, they're not getting much sleep at night. They're in the lab. They're trying to figure out in the cell, why is collagen getting produced? Why is the fibrosis getting produced from certain types of cells? Then there's also animal studies. You can get what we hope to have and what we don't have yet in pulmonary fibrosis. And this is one of the hurdles why we don't have really good medicines. We don't have a good small animal, like a small mouse or a small hamster, that develops spontaneous pulmonary fibrosis. If we had that, then we could test 1,000 chemicals all at once and figure out which one slowed it down or stopped it or reversed it or turned it on its tracks. We just don't have that yet. That's one of the holy grails of research, is try to get an animal model so that we can do a lot more research. Then finally, human studies, human repository studies. We have active protocols at UT Southwestern. Dr. Glazer heads up one of them. I head up another one. This is a very active part of our research, and the drug companies now are trying to get into the game. So for you guys who are on IPF Pro and some of these other ones, they're trying to start their own biorepository studies. And then finally, clinical trials. Clinical trials are the best trials to determine whether or not a medicine works in humans. Okay? So I'm going to start with first talking about some of the genetic studies, kind of moving that on to some of the other basic science kind of studies. And Dr. Glazer's done a beautiful job already talking about clinical trials. Okay, so the first thing, genetic studies. What do we mean? We're talking about what genes are encoded, hard-coded in your genome that gives rise to pulmonary fibrosis. And you can think about it in terms of looking for a needle in a haystack. The way I think about it is if you take all the cells in your body and you take all the DNA from all those cells, you've got enough DNA to make a trip from the Earth to the sun and back 10 times. So that's how much coding you have in all the cells in your body, especially if you're thinking about over the lifetime of a person. We have so much DNA. And that DNA is just this string of four bases. It's just one long string. And what we're trying to do is, if you can look at the middle of that magnifying glass, is find the genetic codings for your disease, okay? And so that's kind of the point of genetic studies. And so how do we do that? How could we get over that hurdle of looking through all those bases and find something that's related to your disease? So what we do here at UT Southwestern is we have a whole lab that's devoted to that. And, it starts with actually asking you for a blood sample. So, we talk to you about our genetic study. We, ask you whether or not you'll stick out your arm for us and donate a sample of blood, and that goes to the lab. And the lab then is responsible for isolating that DNA from that blood sample. So that's how all of our studies start, okay? Then we have a wonderful machine that actually automatically extracts the DNA. There's robots, etc., so it helps with the technician time. And then we can actually start sequencing all that DNA. So this is a big area of research. All that DNA gets put away into refrigerators and freezers and in liquid nitrogen freezers, and sometimes we save your cells so we can go back to your cells. Okay. Then after that, this is where the big developments have occurred, I would say, in the last 15 years. We can now sequence your DNA in ways that we had never thought possible 10 to 15 years ago. That's why it's called the genetic revolution. That's why the buzzword now is precision medicine. Because for basically in the lab, for about $1,000, depending on how well you want it sequenced, between $1,000 and $5,000, we can sequence every single base of DNA in your genome. So then we take all that information and we want to know, well, what are the genes that cause your disease? All that data gets stored. We've got servers and computers and rooms full of computers and servers storing all that DNA data. And then we have to figure out with that data, well, where are the genes? Okay, so I'm going to give you two examples of how we do that. One is we use the power of families, and Heather Cagle already mentioned this. This is one of the main strong points for genetics. We basically go to families. We say we would love it if everyone in your family could help us, and we basically collect blood from everybody. We collect the data from everyone, and then we're comparing what do the people with pulmonary fibrosis all have in common. Now, fortunately, most of you with a family history only have like one or two people who have pulmonary fibrosis. But there are some unfortunate families who have been referred for the research that have, there's one I have that has had 20 plus people in their family with pulmonary fibrosis. So very, very strong kind of a genetic predisposition for disease. And those are the ones that really are the most informative. Fortunately, they're extremely rare families. But those are the families that are the most informative for really narrowing it down and what causes the gene for the disease. So we sequence all of it and we compare those that are affected versus non-affected. Now, I'm going to give you one example. This is an old example, but this is an example of a family that was referred to me by Fernando Torres, who runs the transplant clinic. So in all of these studies, squares mean men in a pedigree, and circles mean women. So you guys are going to be geneticists, so you're going to be able to read a pedigree. And the arrow means that's the person that first came to medical attention. And if you can see down here, I don't know if you can see my pointer, maybe not. But there's a square there and with an arrow. And that was the one who went for lung transplant. And when Dr. Torres talked to the patient, he said, well, my brother died of pulmonary fibrosis. He also had some rare type of lung cancer, and my mother actually died as well. And so Dr. Torres, he has my cell phone, he just calls me right then and there, he says, come over to clinic right now. So my staff or someone goes over there. And when we start talking to the family, and this is no slide on the gentlemen in this audience, gentlemen know a slice of their family history, but what we really need, we need to talk to the women of the family. Because when we talk to the women of the family, this is what we got. So they knew, because women tend to stay in touch, and it turns out that one generation above, there was more than 10 kids, and they kept in touch with cousins and second cousins, etc. And so they pointed us to contact individuals. They made the first phone calls, because of course with HIPAA, I can't call anyone. I can't cold call anybody. So it was really the women in the family who were calling all the relatives. Could Dr. Garcia call you? Can you give them a sample of blood? This is important. So we did all of this. We did that, what I told you about. We sequenced a bunch of DNA samples, and we basically ended up with a candidate gene. This is a gene that encodes for the most abundant protein in the lung. So it was obviously a good candidate. It's basically only expressed in the lung, and it consists of the most abundant protein that's secreted out from the lungs, epithelial, or the cells in the lung, and protects your lung from the environment. And so we do know that some rare cases of familial pulmonary fibrosis are due to mutations in this gene, and this has been replicated now in a French group and also by a group in the Netherlands. Okay, so what do we do with that information? And so this is where, again, technology is really just booming ahead and helping with this research. Because I would say in the last 3 years, there's a new technology that's developed now, and it's called CRISPR-Cas9. And it's a way, think of it as a very fine pair of scissors. You can actually go into the genome of a mouse and change one base. That is one base out of 3 times 10 to the 9th bases. You can go in and very specifically introduce one change using this technology. So what we've done is we now have a mouse that has that family's mutation in that one gene. And so we're very excited because we just basically had this breakthrough to develop that mouse, and now we're expanding that mouse colony. And we now know in the mice that the mice can have one bad copy of the gene. That's what all the people with pulmonary fibrosis in our human family had, only one bad copy of that gene. But you have 2 copies, right? You have a wild type form and a bad copy. The mice, actually, we can actually breed them so that they have 2 bad copies of the gene. So we've just done that, and now we're trying to figure out, well, what's going on? How does this teach us something about pulmonary fibrosis? And I have to say, I don't know. I'm showing you data that is literally hot off the presses, okay? I have no idea. I've got a lot of people working in the lab, and we are working on this really hard and fast, and hopefully we'll have some answers for you. But this is, I'm only going to show you one piece of data because I'm just so excited about this. Dr. Glazer doesn't even know about this. This is literally just this week's data, where we went through and we basically took these mice, and then we collected that fluid secreted from their lung, and we also harvested the mouse lungs. And we wanted to know what's going on with this protein. So in the wild type mice, we can see that they're making the protein just fine, right here. The mice that have one mutation actually have just a little bit of the wild type protein. They have this abnormal protein. And then the mice with the 2 bad copies of the gene, looks like they really don't have much protein at all. And in fact, when you look at the lavage to see what's secreted, they don't secrete any of this protein at all. And the one with one mutation basically has a big hit in the amount of protein that gets secreted. So we don't know exactly what this is going to do in terms of fibrosis. We're trying to get these mice, see if we can get some scarring going on, see if they'll develop pulmonary fibrosis. We're doing a lot of things to these mice now, see if we can get an animal model. But this is basically an example of what we're doing with the genetic work that starts in humans. That's all of our research starts with humans, and we're going to try to figure out if we can get an animal model so that we can test new drugs. So that's one example, okay? Okay, another example. So that was using families. Now, we can also, so a lot of you may have been approached by myself, Dr. Glazer, one of the other physicians in the clinic, or by Tion, saying, would you want to participate in our genetics research? And for that, we take people who have a family history. We also take people who don't have a family history. And the thought process is, if we can just get enough people with pulmonary fibrosis, maybe we can really expound on terms of our discoveries of what causes pulmonary fibrosis. Maybe not just in those families, but in the population at large. And so we've done that, and a number of other groups have done that. And again, I'm just going to give you one example. So if we sequence everyone with pulmonary fibrosis, we collect all that data, then we sequence a whole bunch of people who don't have pulmonary fibrosis, our controls that we're collecting. So if we have a bunch of people on both sides, now when you look across all those billions of variants, can you find some that you find just more common in those with pulmonary fibrosis? And the answer is yes. The statistics is very complicated. That approach by itself basically led to the discovery of 2 genes that we published 2 years ago. And those 2 genes are named PARN and RTEL. They're actually in the same pathway of other genes that we had discovered earlier, and that's in the telomere pathway. And I just want to give you an interesting side note. When we started this study, these were actually those with familial pulmonary fibrosis. But when we were looking at all these variants in the billions and billions of genetic variants that we were collecting, we found one variant that had never been found before in the world's populations. And this all genetic data basically gets deposited in databases to help genetics at large. So if you look at the world's largest database, now this is including almost over 100,000 people collected across the world, we never found this variant. So it was what we call a novel, completely new. And yet when we looked in our database, so the local database here where we had collected DNA samples from you guys, we found it twice. I was like, well, that's odd. It's never been seen in the world, and yet we see it twice in Texas. Is this a Texas variant? What's going on here? So it was found in one of my patients, and it was found in one of Dr. Fitzgerald's patients. And we called, again, had to talk to the women in the family. Sorry, guys. Turns out they're related. They had lived within 100 miles of each other in East Texas, and they didn't even know it, okay? And I haven't told them, okay, so don't tell them. But it turns out that's how genetics was a huge clue for us, that one, they were related, and two, this variant really is linked to pulmonary fibrosis, because everyone who ended up with pulmonary fibrosis in the collective large family had this variant. But the important thing is that people who had this variant, more than half of them did not end up with pulmonary fibrosis. So even though it's a risk factor, it's not telling us that you will end up with pulmonary fibrosis. And that's really how we think of the genetics of this disease, that it's multifactorial. The genetics is one part of it, right? You're born with your whole genetics, right? And that has to last you for your entire lifetime. But this is a disease that comes on at the latter stages of life. So there's a lot of things that happen in between. And we now know with most of these genetic variants that at the most, maybe 50% of people will end up with pulmonary fibrosis. More likely, it's probably more than 50% will not end up with pulmonary fibrosis. And the more careful we are about those environments that our lungs have to breathe, this is why for the young people in the audience, I go on these family draws and I say the one thing you have to hear me say is do not smoke and protect your lungs, okay? Because if you're in a family that has this or has a risk of this, those are things you can start changing now. I can't change your genetics, but those are things that we can change now. Okay, so the familial form is definitely rare. Thank goodness, it's very rare, but we have been making discoveries on a lot of the different genes. In fact, 5 different genes have been discovered here at UT Southwestern. Like I mentioned, actually now more than 4 of these genes are in the telomere pathway. And just to tell you a little bit about that, telomeres are these unique pieces of DNA at the ends of chromosomes. Think of them as those little plastic pieces at the end of your shoelace. So if those little plastic pieces or metal pieces at the ends of your shoelace are intact, basically you can think of your shoes as probably looking like a new pair of shoes. But if those plastic pieces get all degraded and destroyed, they come off, unravel, and your shoelaces unravel, that's kind of what happens to your chromosomes. And your chromosomes, when they start unraveling, some bad things can happen, and we now know that pulmonary fibrosis is one of the physical, clinical manifestations of short telomeres. Okay, so what does this gene do? What does telomerase do? And this is my segue into stem cell biology. So telomerase is an engine, okay? It basically adds these bases at the ends of the chromosome. It lengthens the chromosomes, and when you have long telomeres at the ends of your chromosome, your cells can divide. In fact, they can divide not just once, they can divide many, many times. When those little ends get really short, then that's when the cells can't divide. There's these pathways that get activated that prevent it from dividing. So in cancer is a disease where there's unlimited cell division, right? Those cancer cells just keep dividing. Guess what? Over 95% of cancers, they've taken over this mechanism and they've upregulated telomerase. They've basically taken something good from the human body and they've usurped it so that the cell keeps dividing and dividing and dividing. So cancer cells take telomerase and overexpress it. People with pulmonary fibrosis and other like disorders basically have less telomerase, underexpress it, and those ends get shorter. So I think of it as kind of like a seesaw mechanism. So those with germline or inherited mutations in any one of a number of genes that affect this pathway, all the humans that we know with the genetic studies have one bad copy of the gene. They also have one normal copy, one wild type. But just that slight drop in the activity over time gives them that risk for pulmonary fibrosis. In studies that I'm not going to show you any data that we just published a couple of months ago, we found that that risk in humans was enough to basically drive the acquisition of cancer-associated mutations in the same gene to give the cells that ability to divide even longer, to kind of balance out the seesaw. So that acquisition of basically promoter mutations didn't occur in the germline, but it occurred in other cells, and it kind of balanced out the C-cell. To put it in layman's terms, think of it in terms of a battle going on. If you don't have enough telomerase, your cells are going to stop wanting to divide. If you have too much telomerase, the cells are going to divide, and they're not going to be responsive to the normal signals to say stop dividing, and you get cancer. So there's a seesaw. It's really important for all the cells in our body that this seesaw is maintained at a really good balance point, right? Because we don't want too little or too much. Okay, so what's a stem cell? So this is copied from the dictionary. A stem cell is basically an undifferentiated cell, and it's got the potential to divide and divide and divide, and it has the potential to become a particular cell type. And usually that cell type can be one of a number of cells. It could be a hair cell, it could be a tongue cell, it can be a lung cell, a leg cell, etc. It's got the potential to be multiple different types of cells, but more importantly, it can divide many times so it can generate that cell. Okay, so then I just Google search, okay? I figure, okay, you guys, if we throw out a term, the first thing you're probably going to do when you go home is you're going to type it into Google, and you're going to start going down. What's hit 1, hit 2, hit 3? So these are the top 3 hits, okay? When you type in stem cell and IPF, okay? First of all, there's an ad for a company. It's your number 1 hit that says, come to us. We can basically extract some cells. We'll make them into stem cells. We'll put them back in your body. And they're going to cure everything, okay? That's the first hit. Second hit is actually a news brief from the Pulmonary Fibrosis News that says, finally, at last, the FDA is turning its attention to stem cells for therapies like pulmonary fibrosis and other chronic ills. Third hit is actually a letter that came out, put out by Greg Cosgrove, I think there's another author as well, from the Pulmonary Fibrosis Foundation, because so many people were approaching them and asking them, what about stem cells for my pulmonary fibrosis? And if you haven't read the letter, I encourage you to go online, read the letter. There's since then been a number of published letters, even in the New England Journal, which is one of the top, medical journals about stem cells for pulmonary fibrosis, for, knee pains, for cartilage problems, for heart dysfunction, etc. And they've also put out a a statement as well. So what is the answer, okay? First, I'm going to say a word of caution, okay? I've told you about sequencing the genome. I've told you about this great technology called CRISPR-Cas9. Those are existing right now. The technology for making stem cells does not exist, okay? So I just want to caution you, if it doesn't exist, buyer beware. Be careful what you're going to buy if someone says, I'm going to sell you stem cells, because I'm telling you as a scientist, it doesn't exist. We just had Rudolf Janisch give the lecture on Wednesday. He's one of the world-class leaders in stem cells from MIT. And he basically, his whole lecture was all about the impediments for getting cells to act like stem cells. And they're working in mice. They're not even working in humans. They are working in mice, and they're trying to get it to work in mice. So it doesn't yet exist. Now, I am hopeful this is going to exist at some point. I'm hopeful that in certain therapies will come out of the research that's getting done for stem cells, but it just doesn't exist right now. It doesn't exist in a mouse. It doesn't exist in a human, okay? A word of caution for all of you. And Dr. Glazer and I were talking about this a few days ago, and he brought up a great point. You know, when I started my career, it was in the days of one of the first clinical trials, the gamma interferon trial. I don't know if anyone's remembered that term or remember that study. That was one of the first intermune studies. And it hadn't even been studied in people with IPF. And we had patients basically selling, cashing in their entire life savings to get this medicine. Turns out, most of you don't know what gamma interferon is because it didn't work for IPF. Okay, so just again, a word of caution. That's what was going on 15 years ago. Okay, so second of all, the science about stem cells is that they have to live in very particular parts of the body. And scientists, we call these their niche. The niche for a stem cell, basically, it's surrounded by what's called nurse cells. They have to be protected. They have to get nutrients. They have to get a blood supply. They have to live in close proximity with multiple cells that take care of it. Just think of a baby and a mother and that kind of intimate bond. A stem cell has the same relationship with the nurse cells all around it, okay? Because it has to stay in that protected, undifferentiated state before it's allowed to differentiate. And for most examples in the human body, the stem cell is living in some proximity in the organ in which it can repopulate. So a lot of work has been done in the bone marrow. The stem cell in the bone marrow is in a certain area, nurse cells all around it. The stem cells in the GI tract that basically repopulate your whole gut after you have a horrific case of diarrhea, those are in a specific niche at the bottom of the crypt around all these cells that take care of it. The stem cells in the skin, after you have a big sunburn and you have to repopulate your skin or you have a burn, they're in a particular niche down deep in the basal level of the skin. Those are just 3 examples, but in general, it's in a niche. In the lung, we don't yet know exactly what the niche is. And the mouse tells us there's multiple different niches for different stem cells for different parts of the lung. Like the large airway has a certain stem cell. The twigs, the small airways have a different stem cell. The leaves, the distal part, the alveoli have a different stem cell. That's what the mouse stem cells tell us. And so, so the science behind that and how to keep their stemness is really important. And it's great, fruitful research, but we just don't know in the human yet. There's some really interesting studies that's coming out in the human about where that niche is for the distal lung. And I'm anxious to see how that's going to play out. Third point is this balance of regeneration and cancer. Stem cells, remember, they can repopulate. If you burn your skin, they can repopulate your skin. The skin forms where it needs to form. It doesn't start overgrowing your skin. It doesn't start growing other places, right? It knows, it has a memory. It knows where it needs to go, and then it stops growing. That's an amazing feat of biology. But what cancer does, it uses the same pathways of regeneration, but it's lost the signals to tell it to stop. It keeps going. Cancers don't respect boundaries. They just keep on growing and growing and growing, okay? So the difficulty of playing with stem cells is that you may know what the pathways are, but how do you tell that cell, how do you give it the programming to know when to stop? And there was a case that we just heard about on Friday of a patient who had stem cell therapy in, I think somewhere, I can't remember which country. Anyway, they came in CUH, and they presented with this incredibly rapidly growing cancer that affected their lungs. And of course, we're not going to be able to say, oh, it was definitely the stem cell therapy, but, due to the biology of this, we don't know yet. So it's kind of like playing with fire. And then finally, clinical trials. So clinical trials are the gold standard for any treatment before it's helpful in humans. We need to have good clinical trials. If it's a therapy that people say works, I would say, show me the data. That's what I tell my lab. Show me the data. Show me that it does work. And as a patient, I would say, show me the clinical trial where it absolutely works. Okay, with that, that's my little soapbox on stem cells. I just want to thank you for your attention. This is a great group. We want to support this group in any way, shape, or form that we can. We're so thankful for you. We're thankful for the partnership that we have with you. And I'll just open it up to questions then. I'd be happy to take any questions. Yes.
I was wondering if you about of the University of Colorado. My husband's in a study there where he presented lung problems long before his rheumatoid arthritis, and years. And they had a theory that a drug, bacteria, or protein gets into your lung and begins to cook in there, and at some point it causes that inflammatory response. And then they got real interested in bacteria in your gum line. And he's so Scottish descent, he's got big bad teeth. So anyway, they got real interested in that showing up in other people with this, Are you familiar with that work, or is that anything that you all are also working on?
Right. So part of that registry that Dr. Glazer basically spearheads is a registry where you assign your name and you allow us to track your medical history in the medical records. Even we can track it without your, as long as you give us that permission, we can track it over time. So, we have, and I should say that in the combined registry between both of us, we have over almost 4,000 patients enrolled and over 1,000 people with different forms of pulmonary fibrosis. So one of the projects that we've given to one of the fellows, so these are up-and-coming lung doctors, is figuring out this relationship between autoimmune disease and pulmonary fibrosis. And this guy's really brought some interesting data to my attention. One, the lung disease can precede the onset of the diagnosis of an autoimmune condition. We estimate that it may be 10%, maybe a little less than that. That is the case. We don't yet know. I think it's intriguing that it could be a bacteria. There's some trigger, right? We definitely know there's a trigger, but we haven't identified the exact nature of what that trigger might be. And so, I think it's fascinating, but those are questions that we're trying to answer here. But, a lot of people are trying to answer that and find out what the trigger is. Because if we could figure out the trigger, then we suppress the trigger and hopefully, it would never flare up. So.
It seems to be in this family.
Okay.
It seems to be in this family.
Yes, ma'am.
I have a stem cell question. A stem cell then cannot become any cell? Like if skin stem cell is only for the skin and not a bone? I mean, you can't make it anything you want then.
Okay, so there's different definitions of different types of stem cells. I'm glad you've asked this question. So the best studied ones are the ones of the stem cells for the organ in which it's going to repopulate. But there are undifferentiated stem cells that theoretically have the potential to become any kind of cell in the body. And those are cells that are basically ones that we call embryonic stem cells. So they resemble the embryo in its earliest stage, the 2-cell, 4-cell, 8-cell stage, when you existed at just a stage of 4 cells. We do know in the mice that the embryonic stem cells do have that potential to differentiate into multiple different tissues. But that gets quickly shut off. And so what Dr. Yanish was talking about is about those studies where you could take, say, a skin cell and reprogram it. So the scientists who figure out how to reprogram cells into more like an embryonic stem cell won the Nobel Prize. It turns out it needs 4 genes, needs these certain environments, etc. But even using that technology, the Yanish lab cannot get those cells to truly act like an embryonic stem cell. They can't mix that population of cells in an embryo and show that it can mix and form a chimer. There's some block for those where you're taking a truly differentiated cells and trying to get them to go all the way back. So a lot of the lecture was, what is the block? What are, how can we bypass the block? What are the genes up or down, etc. So as of this point, it's just very complicated. Okay. Yes, great question though. Yes, another question.
It was mentioned that there are various triggers that cause different types of pulmonary diseases, and like asbestos, birds. Is there a list someplace that lists some of the things, like maybe if you have an allergy to something? Is that something that can cause these conditions?
That's a great question. So the question is, what are the triggers? And I wish I knew all the triggers. There are lists of things that you can inhale that are associated with fibrosis. So I call those fibrogenic insults, or things that you can be exposed to that can give rise or has been associated in the literature with fibrosis. The list is incredibly long. Actually, Dr. Glazer is one of the world's experts on that. And there are all the review articles that I read, that he writes and his colleagues write. Usually the table lasts like 3 pages.
A list of 200 of them are on the PF Warrior site on Facebook.
Great.
Okay,
Perfect.
thank you.
But I want to say that smoking's number 1, and then all the rest are and birds are probably number 2. And then I usually tell my patients, metal fumes, wood dust, feathers, anything bird related, and then kind of a lot more are further down on that list. But yes, great resource for PF warriors. Yes, another question.
Are those things like the birds and the feathers, whatever, is this something that you could be exposed to over time and would finally show up? Or once you're exposed to it, it's there?
I'm going to let the expert tell us.
So it's a hard question to answer because everyone's different, so everyone acts different. But in general, we don't, people have the exposure for a while, and then the immune system has to recognize it. So that why the immune system decides to recognize it in a minority of people that have that exposure, but not in the rest, we don't know. There's lots of studies where people have had a bird for 10 or 15 years with no problem, and then they start getting the disease. So there's lots of theories. Doctors love to blame viruses when we don't have another explanation. So you'll find lots of stuff about that, but it all means that we just don't know.
So what about push down pillows and blankets?
They cause it.
In the bird exposure, initially is that hypersensitivity pneumonitis, or
Yeah, that's right.
and then it has some clinical symptoms, or silent? That's
Well, I mean, who knows if it's silent, right? Because you wouldn't come to medical attention to know. But yeah, eventually, if you get the disease from the bird, then you'll eventually start having some symptoms. The question up front was, does it have to be a bird, or can it just be feathers? Those of you that have been with us in clinic, it can just be feathers. Lots of studies, just feathers. I'll personally never forget it, because when I was a fellow in training, we had a very lucky rich gentleman who had probably 5 houses around the world, and never lived in any one place for more than 3 months, because he would then move on to his next house. And he had hypersensitivity pneumonitis, and it was like, well, good Lord, what are we going to find that is going to be common? Because this wasn't just like different states in the U.S., it was like every continent. So what was it? Well, he never went anywhere without his goose down pillow. That's what it was. Now it's easy for me to remember, because I always just have a picture of that gentleman in my head with that.
Something like the goose down pillows, though, is that a known cause of pulmonary fibrosis?
It's
So
a
it
known
wouldn't-
cause of hypersensitivity pneumonitis, which is one of the types of pulmonary fibrosis.
Okay, so it wouldn't be IPF?
No.
Okay. Because I've identified a lot of things in my history that I've been told, like down and so on, but I'm IPF. What's the hope for me?
I mean, it's hard when you have exposures like a pillow and you have a biopsy that looks like IPF. Remember on my slide when I said if there is a possible exposure, just get rid of it even if we don't know for sure? That's an example. So studies have shown that 9 out of 10 times that happens, there'll be something on a biopsy, if you can have a biopsy, that will allow us to differentiate the two. But even 1 out of 10, we're not always going to be able to tell. And as of now, we don't have good enough measures of the immune system's response available for us to prove it. There are some exposures that in research that are clinically available here. There's in other parts of the world they do clinically do this, looking to see if the arm of the immune system that's involved in hypersensitivity pneumonitis, which is different than the hay fever arm, is reactive to birds. So if we had that test commercially available, then that could help us sort it out. In the meantime, if we can't tell, you get rid of the exposure because that's the most important thing. There was a question back there.
Can it be, I have laryngitis, I'm sorry, kind of as simple as when you're a child, you sleep on your grandmother's feather beds, and you have parakeets, and your dad's a smoker in the house, and then later in life, like when you hit 60, it hits you. Could it be, or do you have to have that your whole life exposed?
Or are you talking about fibrosis in general, or about the fibrosis that you can get from birds?
Well, in general, I guess, because I have IPF, but,
Smoking definitely, individual smoking definitely increases the risk of developing IPF down the road. Because that's an exposure that leads to lung damage, that forces your cells to divide, and if your telomeres are short, every time you divide, they can get shorter. So yes, that can definitely contribute.
well, you don't smoke yourself, but
I was sorry.
someone in the...
We don't really know, okay? We really don't know. For most of those associations of something that you're exposed to, we take a really good history for like the years right before we see you in clinic. We're not really, except for Dr. Glazer, taking that really thorough history, 20, 30, 40, 50, 60 years before. And so collectively as doctors, if we don't have a really good measure and marker of that, it's hard for us to do the studies to say, yes, it's tied. So everyone who slept on a down pillow, their risk is up. You know what I'm saying? So it's just hard to study because the exposure is so remote. But because there's good data in terms of the proximal exposure, that's why we recommend just get rid of everything that's on the fibrogenic list.
In terms of the specific disease, just so you know, hypersensitivity pneumonitis, if you had a bird 30 years ago and you haven't had feather exposure since, it didn't cause that disease. Whether it could have been contributed to fibrosis in the end, that is what we don't know, but it definitely didn't cause that disease. You have to be in that exposure at the time the disease starts for us to think that exposure is related. And the reason for that is, unlike one of you mentioned asbestos. So asbestos, if it gets in your lungs, it's never getting out. That's just the way it is with asbestos, okay? But that's because your body can't break it down. But the feather, it's biological protein. Your body will break that down. It's not going to still be there to cause the problem and allow your immune system to respond to it years later like that. Yes, ma'am.
Drug related to IPF. Are any drugs that you've taken in the past, like Crestor or Lipitor, associated with any studies done on any?
So I think we're getting into that terminology problem there with that question, right? So don't forget what the I means in IPF. Okay?
that's idiopathic. I always say that means the doctors are the idiots and we don't know what caused it, okay? But before we say it's IPF, we look at those things like the drugs and stuff like that. Because if it was caused by the drug, it's by definition not IPF.
Is FPF a form of IPF?
FPF?
Yes.
Familial?
Is familial a form of IPF?
And can you have both?
Yeah. So the world's leaders have not taken a stand. They decided that question was too hard, okay? So what we do is we consider them separate, and I think most people consider them separate. So if you've had one other family member who has pulmonary fibrosis, you fall into that category of familial pulmonary fibrosis. But that's just a description of your family history. And then I try to provide an actual description of the disease process itself. So I provide a clinical diagnosis, or Glazer provides a clinical diagnosis. We know you can have familial disease, but you can have IPF. Or you can have familial disease, and you can have hypersensitivity pneumonitis. You have familial disease, and you can have something else, okay? So we try to provide both terms.
And I know in my family, rheumatoid arthritis, there's a strain, my grandfather had it, my niece has it. But I don't know of anyone else that has the scarring in the lungs. But because of the RA, is that be a strain that would point to what happened?
Yes, so there's clearly genetic factors that can give rise to multiple people in the family having RA or lupus or any number of autoimmune diseases. It looks like there is separate genetics for autoimmunity and separate genetics for pulmonary fibrosis, but then there's also overlap. So it gets really complicated. But yes, unfortunately there's genetic factors for both of those. Great questions. What a great audience.
I know.
What about the therapy for IPF and ILD related RA? Are they similar? And another question along that same line is interstitial, or no, usual interstitial motility pattern that's similar between IPF and ILD related RA.
Okay, so there was two pieces of that. They were both related to RA for those that couldn't hear in the back. So number one is the pathologic pattern in the lung on a biopsy, if we did that, the same in RA and IPF? And the second is what about the treatment? So the answer to the first of those, which I think I reversed the order in the way you said it, if that's okay. The answer to the first of those is most of the time. Okay, it is UIP. Not all the time, because our RA can cause pretty much every single interstitial pattern that exists. RA can cause, but the most common thing it causes is UIP. Okay. The answer to the second question is we don't know. Right now, we still tend to treat RA-related disease the way we treat the other autoimmune-related diseases, which are those other drugs that I showed. But there are active studies on what about using the IPF drugs in those conditions, including RA.
There's one that we're trying to get.
Right, there's one that's not specific for RA. It's any type of pulmonary fibrosis that is not IPF or scleroderma, because those have specific studies that so it's looking at whether or not nintedanib will be effective in any of the other types of fibrosis, including RA.
I want you to take a moment for me and look around this room.
Sorry?
Look around the room.
Oh, yeah.
And think about the continental breakfast we had. And think about two world-class pulmonologists. Here, UT Southwestern is wonderful. They provide the breakfast, our meeting room, this incredibly knowledgeable staff. I want to say thank you to them. They graciously offered to stick around a little while. and answer some questions.
Before we start moving, I was a little late, so the other thing we provide is valet tags. So if you hadn't grabbed one, Patty has one, okay? So that you don't have to pay for the valet.
We don't have one.
Isn't that awesome?
You're here?